Hi everyone, I am Malia Machado and today I will be talking about the characterization of marine viruses from coastal environments within the Central California current ecosystem. So to start off, viruses are one of the most abundant life forms on the planet, yet due to their vast diversity, many marine viruses are still unknown. This study aims to characterize marine viruses that are involved in ecological processes such as carbon cycling and disease pathways. Understanding the interactions between bacteria and viruses is crucial in understanding population dynamics and maintaining ocean environments as major carbon sinks. Bacteria are responsible for between 6 and 26% of photosynthetically fixed carbon being regenerated and available for other organisms to take back up. This is important not only for our ocean systems, but also for our planet as a whole. Our specific research objectives are to identify viruses present in marine environments from marine water samples using sequencing, then to taxonomically classify viral sequences and determine their function or functions within marine viral organisms, and finally to assess quality of viral sequencing data and analysis pipeline for samples collected off of Point Reyes, California. So to do this, viral samples were collected from the NOAA Fisheries Survey vessel, the Reuben Lasker, during the 2017 Rockfish Recruitment and Ecosystems Assessment Survey, where eDNA and bacterial samples were also collected to further look at the communities associated with these samples. However, this portion of the study focused specifically on marine viruses. Collections occurred on May 1, 2017 off the coast of Point Reyes, California. Samples were collected at a depth of 10 meters using Niskin bottles, and so to target and isolate viruses from seawater samples, each sample went through a tangential flow filtration and cesium chloride gradient purification process. The DNA extractions were performed on these filtered samples from Station 1046 and Station 165, as denoted by the yellow stars in this figure. This is a visual that walks through the overall workflow for the sequencing and analytical portion of this project. Sequencing was carried out using Oxford Nanopore technology MinION platform. This protocol includes N-prep and DNA repair, adapter ligation, bead cleanup, and fungal preparation. The sequencing results took the raw reads and overlapped them to form a large genome or metagenomic assembly using the program MetaFly. Quality of our assembly was assessed by using the polishing tool that could detect minor errors and/or gaps. We did polishing and unpolished reads and assemblies to see whether or not there was a difference in outputs and quality. Predictions of possible genes or proteins in the resulting genomes were done using GeneMark S. Here, we were trying different GeneMark algorithms or parameters to see if they yielded different results because they were based on different databases of existing genomic information that could maybe influence our overall gene identification results. To assess quality of assemblies, reads were aligned to the assembled genome using Minimap2 and examined visually using IGV. We blasted our assembly to see if we could identify or associate it with something else that we have already established, some other known product, by looking at other things that have been sequenced in the past. Taxonomic classifications of all sequences were performed using Kraken for classification and then Krona for visualization in OmicsBox. I would like to note that, to our knowledge, these are unique and novel results and methods that have not been used to characterize marine viruses in the past. So we are creating this workflow as we go, and we are trying different methods and approaches. So samples from Station 165 were able to generate assemblies successfully, which means they had enough isolated DNA to create these assemblies. Samples from Station 1046 were unable to be assembled due to the vast amount of disjointiques, which is DNA sequences that do not overlap in any way, even though more data was actually generated overall for this run. This suggests that the samples from station 1046 are composed of a wide diversity of species instead of being composed of a greater concentration of just a few species, making it hard to construct one continuous genomic assembly. This table illustrates the overall coverage for each assembly. The coverage refers to the number of unique reads that call the same nucleotide for a particular sequencing region. This is an example of one of our assemblies in IGV, the bottom, showing the coverage of the assembly. The 54x coverage means that each base was called, on average, 54 unique times for that specific nucleotide in the assembly. The unpolished assemblies were not included in this table because they yielded the same results as the polished versions in regards to length and coverage. However, coverage differed between 165A, 165B, and 165A and B combined, where 165A and B combined had the most coverage with this 54x coverage. Additionally, when we looked at the line sequences of 165A, 165B, and both of them combined, there were very few differences between them across all genomic assemblies, meaning that all of the pieces of the genomes aligned to each other, and it looked like they were circular in nature, and that they had constructed a complete and well-assembled genome. This table shows the same raw and polished assemblies and shows the different outputs we get when using different gene mark parameters. As we can see here, there's little variation between polished and unpolished reads, but there is a difference in the amount of genes predicted using the different gene mark algorithms or parameters from 1999 versus 2010. We ended up using the 2010 version for the rest of our analysis because this used a newer database version. These pie charts created from Krona show the percent makeup in each of our samples where we were focusing on considering only potentially viral sequences. Basically, after trying to create really well-constructed assemblies, we wanted to filter out anything that might be human or bacterial and only keep the parts that were characterized as viral or uncharacterized. Uncharacterized meaning that these sequences did not match any sequences in the database we were using. The same assemblies were generated even after human and bacterial DNA were filtered out, confirming high-quality assemblies. Krona analysis resulted in 0.6% of our sample being classified as viral DNA for 165A, 0.2% for 165B, 0.2% for 1046A, and 0.1% for 1046B. So based on these findings, a lot of what we had in our sample is still uncharacterized and unknown. This figure illustrates our best BLAST hits, showing the most common sequences that match specific known organisms. As you can see here, most of them are bacterial and viral, and there's a range of percent similarity between all the observed. The bottom left graph in green illustrates that out of the matches we did generate, the actual hits were on average only 60% similar, which is not very similar. In addition, the high score segment pair, the HSP, divided by the hit coverage distribution graph in the lower right, illustrates that there was a few hits that matched the entire sequence segment that they were hitting to, but most of them only matched small regions of the hit that they were matching to. This further emphasizes that our results did not match that closely to that of known organisms in the database. The top table is an example of some of the data that was generated for 165A and B combined once it was blasted and compared to already known sequences, mapped to these sequences for alignment, and then annotated, meaning that the function of that sequence segment was determined. So after all of this was done, we only got 2 final annotations back, and they mapped to mostly bacteria and some viruses. Many of these annotations were actually hypothetical proteins, which means that their functionality has not been determined yet. So explanations regarding our findings suggest that there was either contamination in our samples. However, there is evidence against this because we had well-covered assemblies and a lack of discontinuities within them, Or our databases could have been biased and prone towards classifying bacterial characterization and function purely due to more bacteria information being available in these databases. We also could just have a unique sequence because even the matches we did find were not very closely matched. And this suggests that this may be a new sequence that has not been classified or characterized. And finally, we know that bacteria and phages intermix genomes because of their reproductive cycles, so they may not adhere or conform to our perfect genome transfer models, making them very difficult to analyze. To our knowledge, this is the first study to investigate viral diversity from marine water samples off of Point Reyes, California, using the novel methods and approaches with Oxford nanopore technology. Here, we generated a novel genome sequence, which may be a previously undescribed marine virus. Based on our sampling methods and annotation results, we believe it to be a bacteriophage. The field of marine virology is still very young, and many potential viruses are unable to be properly classified due to the current limit composite genome references that are available. This field of research is important to better understand how viruses affect marine populations and overall ocean health. In the future, we plan to continue using analytical tools to determine the unclassified sequences in our samples. In conclusion, there is a lot that was challenging about this process in extracting viral DNA from water samples. However, we may have found a sequence for a virus that previously has not been defined, which is pretty cool. We hope that this study will help bring to light the importance of marine viral research and help further conversations within the scientific community surrounding newly evolving approaches and analytical techniques and tools. Lastly, we would like to thank the NOAA Center for Coastal and Marine Ecosystems program for funding this project. Additionally, we would like to thank Lily Lee for her help and contributions during the sequencing process. We would also like to acknowledge the National Oceanic and Atmospheric Administration, National Marine Fisheries Service, who conducted the annual rockfish recruitment and ecosystem assessment survey. And finally, we would like to acknowledge the crew aboard the fisheries survey vessel, the Reuben Lasker, from which the samples were collected. We are so excited to share this research with you. Thank you so much for listening.